A recent requirement in life science is the observation of biological processes in the context of in vivo systems. Although fluorescent microscopy techniques have developed significantly over the last decades, scientists are still limited either by the speed or by the quality of resolution when imaging 3D specimens. The spinning disc confocal system brings new possibilities, making imaging more effective compared with the widely used laser scanning confocal system. For the time SDC needs to acquire a stack of 22 images for 2 scans, LSM only gets 2 images. What makes the SD system faster is the Yokogawa Confocal Scanner Unit. It is arranged by 2 coaxially aligned discs with nested spirals of thousands of pinholes and a dichromatic mirror positioned in between. Yokogawa improved the illumination of the specimen up to 70% by adding microlenses to the top of the pinholes in the upper disc. Each 30-degree disc rotation produces an image. By regulating the disc rotation speed, we can acquire up to 2,000 frames per second by multi-point scanning instead of the single point at a time used in LSM. This scanning principle makes new things possible for researchers.
So now we can image samples live, so live cells, through thick samples or relatively thick samples at high speed that we were not able to do before. So before, if we wanted to do live cell imaging, for example, in 3D microenvironments, then using the normal confocal, we would not be able to do long time lapse because we would kill the cells quite rapidly.
With SDC, Guillaume images the migration of cancer cells on cell-derived matrices to study the importance of protrusions for the invasive properties of cells. Apart from the scanning unit, the detection of signals by cameras plays a major role in the productivity of the SDC system. It is based on CCD cameras, which have better quantum efficiency than photomultiplier tubes used in LSM.
The system what we have, there is 2 cameras. One EMCCD camera for live cell imaging, and then we have a scientific CMOS camera, which has the frame of 2048 to 2048, and it can really capture really wide field of view. So cameras, they give good signal to noise ratio, so by that we can use like less light if needed, and still we get the nice signal.
All these features make the SDC system user-friendly, allowing less time on adjusting settings and acquiring the images without compromising the quality of the images.
For example, whole mount samples that some research groups want to image here, they are really easy to image now. Before you had to spend a lot of time to do the scan, like montage image, if you want to image multiple tiles and put that together. With the normal samples it took like 45 minutes even sometimes, but now it's just like 10-5 minutes.
This microscope is turning into a working horse at the cell imaging core, presenting a solution for different biological applications.
So at the moment we are starting to do imaging, for example, in zebrafish embryos or in a brain slice, that to do it live on the confocal would have not been possible.